It's a great pleasure to introduce Daniel Grimes, who will be talking about linking cilia motility and cerebrospinal fluid flow defects to the etiopathogenesis of idiopathic scoliosis.
Okay, thanks a lot. So it is a pleasure for me to be here today. One moment. Okay, thanks. Okay, I think we're sorted now. So I'm a postdoc in the Bedine lab in Princeton, and I want to say right at the start that the project I'm going to describe today has been a collaboration between myself, another graduate student in the Bedine lab, Nicholas Moranti, and also a close collaboration with the Saruna lab in Canada, in particular his student, Curtis Boswell. So in the Bedine lab, we're interested in the biology of motile cilia. These are relatively short cellular projections which beat or rotate. You can see in the top left corner, I think, some cilia on chlamydomonas. Now, in single-celled organisms, motion of cilia can propel the cell through fluid. But in multicellular organisms, cilia typically adorn the surface of specialized cells. You can see on the lower panel the Xenopus larval skin with these patches of multiciliated cells. Now, on these cells, the cilia beat in a coordinate fashion, and they can drive the flow of fluid across the cell surface. So the project I want to tell you about today, for us, became interesting via forward genetics. So we had a mutant in the lab called curly, which has very striking phenotypes. You can see here that in mutants, we have a curly tail down phenotype during embryogenesis. So this is about 3 days old. It also has phenotypes that you can't quite as readily see here, like kidney cysts, and also left-right patterning disorders. So this mutation affects cilia motility, not cilia structure. So the cilia form correctly, but they don't beat. We did genetic mapping and found that the mutation affected a gene called C21 or 59. This is a protein previously implicated in cilia biology by Ian Drummond and also other people working with chlamydomonas. But we were particularly interested by one of our mutations, I44N, because it turned out to be temperature sensitive. So that you can see in the middle panel at 25 degrees C, a lower permissive temperature. We didn't see defects. We saw normal cilia beating and no embryonic defects. But at 28 degrees C in the bottom panel, the cilia stopped beating, and you see the profound curved tail. So this temperature sensitivity was really, for us, an opportunity to shift the temperatures at which these fish are raised so that we could rescue early embryonic defects by keeping them at a low temperature for the first 5 days of life, and then shift them to a higher temperature to disrupt cilia beating and ask the question, do we see any later post-embryonic defects? And when we did this, we did see quite a striking defect in the overall morphology of the fish, as you can see there. So this is better seen, this phenotype with micro-CT. This is a normal control sibling. You can see the skeleton, in particular, the very straight spinal column. Whereas in our temperature-shifted mutants, we're seeing these very profound three-dimensional spinal curves. Now, the question we had at this point was, is this phenotype specific to the C21 or 59 gene, or is it generally a consequence of loss of cilia motility? So to answer this, we used CRISPR-Cas9 tools to make a mutant in the gene called DYX1C1. This is another gene which the Bedine lab and others have previously implicated in cilia motility. And you can see in kind of the middle panel that the DYX1C1 mutant does have cilia immotility, and it has the expected phenotypes. It has that early curvature of the tail. Now, to assess adult phenotypes in this fish, we weren't able to do temperature shifts. So instead, what we did was to inject at the one-cell stage wild-type RNA for this gene. That provides an early protein product, which allows the fish to overcome any potential early defects, and then we can then grow them to adulthood and assess potential later defects. And in doing this, we again saw these spinal curvatures. And in fact, we did this for two more mutations, CCDC151 and CCDC40, both genes, again, that affect cilia motility but not cilia formation, and we have the same result. So now across 4 different mutant lines, we see this post-embryonic requirement for cilia motility and maintaining a straight spine. Now, the reason this is in a human disease section is that we do think this is strikingly reminiscent of the human disease scoliosis, which is essentially spinal curvatures. Now, there are many ways to categorize scoliosis. The way I've chosen to do so today is to think about it in terms of congenital scoliosis. This is a birth defect, essentially. It's about 15% of cases, and it's caused by vertebral abnormalities. So you can see in the schematic kind of wedge-shaped vertebra or vertebral fusions. And it's quite easy to imagine how these sorts of defects would lead to spinal curvatures. But actually, there's another type of scoliosis, which is generally called idiopathic scoliosis. This is a much higher percentage of cases, something like 65% of total cases. This disease is characterized by the lack of vertebral defects, and also the fact that the curves develop much later in life, during adolescence typically. And interestingly, there's really no underlying basis for this condition. There's not a lot known about it. So one of our first questions was, are our cilia motility mutants, which have these spinal curvatures, are they more reflecting a congenital type disease or an idiopathic type disease? So the first thing we did to look at that, stop working, is to image the vertebra. So you see in that first panel D, we have a control, because it's a mutant at the permissive temperature, and you see the nice repeating patterns of the vertebra. Whereas in panel E, we have a temperature-shifted mutant, but we again see this nice repeating vertebral structure. We don't see any obvious vertebral defects, even though those fish do go on to develop the really striking spinal curvatures you can see below. And I want to very briefly compare this to another fish model. This is something that Saruna Lab published a few years ago now. It's maternal zygotic PTK7 mutants, and this actually is a great model of congenital scoliosis. And hopefully you can see the kind of vertebral defects, the missing vertebra, and the wedge-shaped. So we think our mutants are actually much more like the idiopathic type human disease. We were also able to use temperature shift experiments, owing to the temperature-sensitive nature of the kurly mutation, to try and define when, during development, cilia motility is required to maintain a straight spine. And what we're doing here is starting the fish out at a permissive temperature, such that they don't have any embryonic defects, and then we're gradually shifting them to a restrictive temperature at the times indicated, and then growing them all the way to adulthood, maybe 3 months of age. And you can see that if we shift the temperature at 19 days or 24 days, all of the fish still go on to develop scoliosis or spinal curvatures. This suggests that any times before this are not relevant in terms of this condition occurring. However, this window doesn't go on forever. So you can see in panel D that if we wait until 34 days post-fertilization to shift the temperature, the fish don't tend to develop scoliosis, even though we then keep them for several months at the restrictive temperature. So to us, this suggests there's a defined window when cilia motility is required. It's between about 3 and 5 or 6 weeks, when if you lose cilia motility, your spines begin to curve. So interestingly, at least in our system, this is a period of very rapid growth for the fish. So perhaps, again, it's similar to the human condition. Now, what are the direct consequences of loss of cilia motility? This is where I have to try and play a movie. So can you play the little black box? Oop. Okay, so this is a wild-type brain, and we can see that if we drop fluorescent particles into the ventricle, which is contiguous with the spinal canal, you can see this robust fluid flow towards the posterior. This is cerebrospinal fluid flow. Now, if we look at our model, in our mutants, this is lost. So essentially, the bees just sit around. There's no dramatic motion. So because all of our 4 mutants that I've mentioned so far all show this lack of CSF flow, we think it's the CSF flow itself which is the pertinent thing for driving the spinal curvatures. Now, so far I've only talked about fish. Is this relevant to human disease directly? We approach this by thinking again about PTK7, this gene I mentioned briefly earlier. Now, Brian Saruna's group a few years ago published that variants in this gene are known to cause idiopathic scoliosis. This is an example of one of the patients. And indeed, zygotic PTK7 fish mutants also exhibit a very similar phenotype to what we saw in our cilia mutants. We see late-onset spinal curvatures without vertebral defects. So the question in this project was, are cilia behind this sort of phenotype? We did scanning electron microscopy to look at ependymal cilia. This is what they normally look like. This is the cilia in the brain, covering the entire surface of the ependyma, pretty much, in this brush fashion. In PTK7 mutants, we saw a massive reduction in the number of cilia, and also the cilia that were present were mispolarized. And again, this coincided with complete loss of cerebrospinal fluid flow. So the question then was, is the cilia defect in this case really causing the spine to curve? To try and address that, we used a FOXJ1A promoter element. This is a piece of DNA which drives expression, in this case of GFP, anywhere where there are motile cilia. So this is a motile cilia cell-specific sort of transcriptional driver. And you can see, particularly in the 21 days post-fertilization, you can just about see it, I hope, in the spinal canal. So we used this same element to drive wild-type PTK7, and the question is, can expression of normal PTK7 only in motile ciliated cells rescue the scoliotic phenotype? And the answer was that it can. So you can see the PTK7 mutant with the severe spinal curves, and then the presence of the transgene rescues that phenotype. And as you'd expect, it also fully restores ependymal cilia and CSF flow. So I've told you about 4 different motile cilia mutants, which all lead to loss of CSF flow and late-onset spinal curves. I've also told you about PTK7 mutants, which is a human idiopathic scoliosis loci, which also seem to lose this CSF flow. So our model currently really is that aberrant CSF flow, particularly during this growth phase, causes the spine to curve, and this might be an underlying cause of idiopathic scoliosis. Now, there's a few bits of data out there in the literature which support this. One is that if we inject kaolin into the subarachnoid space, this is a treatment we'd fully expect to prevent CSF flow. You do get scoliosis in rabbits and dogs. And moreover, there were a range of other human conditions, each of which can disrupt CSF flow, and each of which shows a higher incidence than you'd expect of spinal curve scoliosis. So if I've just got enough time, I'll finish with one final piece of data. What we did here is we again used our temperature-sensitive mutant, and this time we're doing the inverse shift. So we're allowing spinal curvatures to start to develop, and then we're shifting them back to a permissive temperature to restart CSF flow. And we're asking the question, can we restore spine straightness to a spine that has begun to curve? And although I think this is definitely an area which can be worked on further, our initial experiments seem to suggest that we can indeed, when the spine begins to curve, as you can see at 18 days post-fertilization on the far right, if we then at that time shift them back to a permissive temperature, we don't see the severe spinal progression that we would. And even if we shift later at 23 days, again, we see very subtle defects. We don't see the severe progression. So we think spine straightness and maintaining a straight spine during growth is a relatively dynamic process which requires CSF flow for that period. And if you do begin to curve, you can potentially re-straighten out if your CSF flow is restarted. So with that, I'll acknowledge all the members of the Bedine Lab, in particular Nicholas Moranti, the grad student that I work with on this. He's actually on the postdoc market. If anyone's interested, please get in touch. And also, again, I'd like to reiterate that this project was a collaboration with the lab of Brian Saruna in Canada and his graduate student, Curtis Boswell. So thank you for listening.
Why and how do you think the loss of CSF flow leads to this curvature phenotype?
Yes, I mean, that's the big question, I guess. Well, we don't know much about scoliosis in terms of even the different tissues that could be involved. So this could be neuronal, this could be neuromuscular. What's downstream of CSF flow and how that leads to severe spinal curvatures is the question we don't currently answer. And there are some cell types in the spinal canal which are known to respond to fluid flow. cerebrospinal fluid contacting neurons, and there are a few others. And I think how they innovate onto things like muscle, for example, could be one way in which loss of CSF flow leads to strange connections or perhaps asymmetrical connections. Potentially then the muscles start erroneously pulling on the spine. And that's just one possibility there. There are many.
Hi, I was wondering when you were determining your critical window for motility, if you controlled for size or developmental stage of the fish, or if you were just looking at days post-fertilization.
Yes, we did do that as well. In the paper it's in more detail, but we use standard length to measure the size of the fish as well as just showing that. Thank you.
Very elegant. Oh, very elegant talk. I really enjoyed that, especially the TS mutant. Great use of TS mutant. The correlation between CSF flow and the phenotype, though, seems to be that. At this stage, it's still a correlation. And one experiment that I think would nail it down a little better is if you tried a tissue-specific rescue, for instance, with a neural tube-specific enhancer, to selectively rescue cilia flow or cilia motility and flow in the spinal canal. Have you tried that?
Yeah, that's a great point. We haven't tried that yet, but it's something we're definitely going to do. Yeah, I mean, of course, the cilia motility genes we've looked at don't necessarily affect cilia structure themselves, but you're right, we need to narrow down exactly which cell type is relevant, because at the moment we just know it's a motile cilia cell type.
Thanks.
Thank you.